Now we are ready again to go in the second part of innate immunity as we discussed earlier. So this will be discussing the second part of innate immunity. Let us go together and enjoy the journey. Cellular component of innate immune system. The three main functions of the cells of the innate immune system are to save the barrier against infection, act as sentinel to detect microbes and damage cells in tissue, and perform effector functions that eliminate microorganisms. Innate immune cells express the virus pattern recognition receptor we have just discussed, and after recognizing the pumps and dumps, they respond by producing inflammatory cytokines and antiviral protein and kill microbes or infected host cells. In addition, some of the cells of innate immunity are critical for stimulating subsequent adaptive immune responses. Epithelial barrier. Intact epithelial surface form physical barrier between the microbes in the external environment and host tissue. The main interface between the environment and mammalian host are skin and mucosal surface of gastrointestinal, respiratory, and genitourinary tracts. These interfaces are lined by a continuous layer of specialized epithelial cells that serve many physiological functions, including preventing the entry of microbes. Loss of integrity for these epithelial layers by trauma or other reasons predisposes an individual to infections. We will summarize the main features of innate defense by epithelial barriers here and discuss epithelial barrier immunity in greater detail in chapter 14. So actually, in the diagram that you will see later on, or you will follow up in the book, there is a physical barrier to infection. Then if that physical barrier is not intact, there is killing of microbes by local producing antibiotics, that is defensin and calthericins, the peptide antibiotics. Then inside again, there is intraepithelial leukocytes, killing of microbes by infected cell by intraepithelial lymphocytes. So this was just a diagram. So the protective function of barrier epithelial is in large part physical. The epithelial cells form tight junction with one another, block passage of microbes between the cells. In the skin, the outer layer of keratin, which accumulate as keratinocyte on the surface, dies, serve to block microbial penetration into deep layer of the epidermis, and cutaneous infections generally occur when there is a barrier breach. Mucus, a viscous secretion containing glycoproteins called mucins, is produced by respiratory, gastrointestinal, and urogenital epithelial cells and physically impair the microbial invasions. The function of this barrier is enhanced by serial action in the bronchial trees and peristalsis in the gut, which facilitate elimination of microbes. Although these physical properties alone are very important in host defense, other epithelial defensive mechanisms have evolved to complement the mechanical barrier. Epithelial cell, as well as some leukocytes, produce peptides that have antimicrobial properties. Two structural distinct families of antimicrobial peptides are defensin and cathelicidins. Defensin are small peptides, 29 to 34 amino acid long, that contain both cationic and hydrophobic regions and 3 intrachains, the sulfide bonds. Two families of human defensin, named as alpha and beta, are distinguished by location of these bonds. Defensin are produced by epithelial cells of mucosal surface and by granule-containing leukocytes, including neutrophil, natural killer cells, and cytotoxic T lymphocytes, CTLs. The set of defensin molecules produced differ among different cell types. Paneth cells within the crypt of small bowel are the major producer of defensin. Paneth cell defensin are sometimes called crypticidin. Their main function is to limit the amount of luminal microbes near the epithelial barrier. Defensin are also produced in the colon, in respiratory mucosa cells, and in the skin. Some defensin are constitutively produced by some of the cells, but their secretion may be enhanced by cytokines and microbial products. In other cells, defensin are produced only in response to cytokine and microbial products. The protective action of defensin include both direct toxicity to microbial, including bacteria, fungi, and enveloped viruses, and activation of the cell involved in inflammatory response to microbes. Defensin kills microbes by a variety of mechanisms, many of which depend on their ability to insert into and disrupt the function of microbial membranes. Cathelicidins produced by neutrophil and barrier epithelial cell in the skin, gastrointestinal tract, and respiratory tract is synthesized as 18 kilodalton precursor protein that is secreted and then proteolytically cleaved into two peptides, each with the protective function. Both precursor synthesis and proteolytic cleavage may be stimulated by inflammatory cytokines and microbial products. The active cathelicidins protect against the infection by multiple mechanisms, including direct toxicity to broad range of microorganisms and the activation of virus response in leukocyte and other cells types that promote eradication of microbes. The C-terminal fragments called LL37 can bind and neutralize LPS, the toxic component of outer wall of gram-negative bacteria that is recognized by toll-like receptor 4. Barrier epithelial contains certain types of lymphocytes, including intra-epithelial T lymphocyte, which recognize and respond to commonly encountered microbes. Intra-epithelial T lymphocytes are present in the epidermis of the skin and in the mucosa epithelium. Various subsets of intra-epithelial lymphocytes are present in different proportions depending on species and tissue location. These subsets are distinguished mainly by the type of antigen receptor they express. Some intraepithelial T lymphocyte expresses the conventional alpha and beta forms of T cell receptor, which is present on most T cell and lymphoid tissue and in circulation. Other T cells in epithelial expresses a form of antigen receptor called alpha delta T cell receptor that may recognize peptide and non-peptide antigen. A characteristic of intraepithelial T cell is the limited diversity of the antigen receptor compared with most T cell in adaptive immune system. These intraepithelial T lymphocytes are thought to recognize small number of commonly encountered microbial structure, a typical feature of innate pattern recognition receptor we have described. It is also possible that these lymphocytes are activated not by antigen recognition, but by cytokines and other molecules produced by epithelial cell in response to stress. Intraepithelial lymphocytes may function in host defense by secreting cytokines, activating phagocytes, and killing infected cells. Now let us discuss phagocytes. Phagocytes, cells that have specialized phagocytic function, primarily macrophage and neutrophil, eliminate microbes that breach the epithelial barrier. We introduced these cells types in chapter 2, and we will discuss many other details of their function in the context of inflammatory response later in this chapter. The essential role that phagocytes play in innate immune defense against microbes is demonstrated by the high rate of bacterial and fungal infection in patients with low blood neutrophil count caused by bone marrow cancer or chemotherapy and irradiation of cancer, which destroy immature cells in bone marrow, and in patients with inherited deficiency in function of neutrophil and macrophage. Neutrophil and monocyte are circulating phagocytes that are recruited in tissue in response to signal generated by innate sentinel cells. The monocyte rapidly differentiate into macrophage after leaving the blood. The tissue-resident macrophage are always present in most tissue under normal conditions and are also saved as sentinel cells. Dendritic cell. Dendritic cells rapidly and efficiently detect invading microbes because of their location in tissue and their expression of numerous pattern recognition receptors for pumps and dumps. Dendritic cells express many different types of toll-like receptor and cytoplasmic pattern recognition receptor, making them versatile sensor of pump and dumps. In response to invading microbes, they secrete inflammatory cytokines that promote recruitment of additional leukocytes from the blood. The plasmacytoid subset of dendritic cell is the major source of antiviral cytokine type 1 interferon produced in response to viral infection. We will discuss the antiviral actions of type 1 interferons in more detail later in the chapter. The ability of dendritic cell to promote T lymphocyte response after innate immune activation also make them as an important bridge between the innate and adaptive immunity. The reaction of dendritic cell to microbes in the early innate response enhances the ability of the dendritic cell to activate T cell in subsequent adaptive immune response. Dendritic cell captures antigen and displays them in T cells. See in chapter 6. Dendritic cells activated by microbial pumps also expresses a membrane molecule called co-stimulator that promote T cell response. In addition, depending on the nature of microbes that induces the innate response, a dendritic cell will derive naive T cell differentiation into distinct type of effector cell, such as interferon alpha, interferon gamma producing T helper 1 cells, or interleukin 17 producing T helper 17 cells. This feature of dendritic cell will be discussed in chapter 9 and chapter 10 because of their tissue resident and ability to rapidly detect infection in the tissue. The dendritic cell are proteolytic sentinel of an immune system. Cytokine producing innate lymphoid cells. Innate lymphoid cells, which were introduced in chapter 2, are bone marrow derived cells with lymphocyte morphology that were discovered as cells that produced the cytokine similar to those made by helper T cell but lacked the T cell receptor. There are different subsets of innate lymphoid cells that arise from the same common lymphoid precursor that give rise to the B and T cells. But the presence of precious steps in innate lymphoid cell development are not fully understood, especially in human. It is clear that during their development, there are branch points giving rise to 3 different helper subsets of innate lymphoid cells, which function mainly by secreting different types of cytokines similar to CD4 helper T cell subset, and a separate branch giving rise to natural killer cells, which function as cytotoxic effector in addition to secreting cytokines interleukin interferon gamma similar to CD8 cytotoxic lymphocyte cell. We will describe cytokine producing innate lymphocyte cells subset in section and natural killer cell in the following section. 3 subsets of innate lymphocyte cells called innate lymphocyte 1, innate lymphocyte cell 2, innate lymphocyte cell 3 produce different cytokine and express different transcription factors analogous to T helper 1, T helper 2, and T helper 17 subset of CD4 T lymphocyte. Innate lymphocyte cell 1 produces interferon gamma and expresses the transcription factor T-bet like T helper 1 cell. Innate lymphocyte C2 cell 2 produces mainly interleukin 5 and interleukin 13 and expresses the transcription factor GATA3 like T helper 2 cells. Innate lymphocyte cell 3 produces interleukin 22 and or interleukin 17 and expresses the transcription factor ROR like T helper 17 cells. Because Innate lymphocyte cells do not express T cell receptor. They must be activated by different mechanisms than helper T cell to produce these cytokines. The best defined stimuli for innate lymphocyte cell cytokine production are other cytokines, sometimes called alarmin, which are released in the context of innate response to infections and tissue damage. Each innate lymphocyte cell subset is activated by different cytokines. The common lymphoid progenitor. Normally we know the innate cell lymphocyte precursor. Then from there, it produces, if it is innate lymphocyte 1, then it is due to stimulation of interleukin 15 and interleukin 7 development. If it is by forming interleukin innate lymphocyte 2, the stimulator is interleukin 7. Then if from letting the interleukin innate lymphocyte 3, the precursor there, our development of that one is caused by interleukin 7. So activation of innate lymphocyte cells, one, it causes production of interferon, the defense against the virus. The activation of innate lymphocyte cell 2, it produces interleukin 5, interleukin 13, the defense against helminth allergic inflammation. Then, if it is innate lymphocyte cell 3, it produces alpha beta 1 and alpha and interleukin 1 beta and interleukin 23, whereby it's also produces interleukin 17 and interleukin 22. They function in intestinal barrier function lymphoid organogenesis. So actually they look similar, almost similar to T helper 1, T helper 2, and T helper 3. Innate lymphocyte cell subset may participate in host defense against distinct pathogen and also may be involved in inflammatory disorder. Innate lymphocyte cells are likely important for defense against intracellular microbes. The innate lymphocyte cell 2 are important for defense against helminthic parasite, and they also may contribute to allergic diseases. Innate lymphocyte 3 are found at mucosa site and participate in defense against extracellular fungal and bacteria, as well as in maintaining the integrity of epithelial barrier. Lymphocyte tissue inducer cells are subset of innate lymphocyte cells, which in addition to secreting interleukin 17 and interleukin 22 also expresses the membrane molecular lymphotoxin and secrete tumor necrotic factor, both of which are required for normal development of lymphoid organs. The feature of innate lymphocyte cell that makes them potentially important for early host defense is that they are always reside in epithelial barrier tissue, poised to react against microbes that breach those barriers. In contrast, T cells circulate through secondary lymphoid organs and migrate into tissue only after they are activated at different and differentiate into effector cells, a process that may take several days after encounter with microbes. Because corresponding subset of innate lymphocyte cells and helper T cell produce similar cytokine, they may work in a temporally coordinated way, with the innate lymphocyte cell being the early innate participant activated by alarmin in infected tissue, and the helper T cells appearing later as part of adaptive immunity. The action of innate lymphocyte cells, natural killer cells, and T helper 1 cells are sometimes grouped under type 1 immunity, along with activated CD8 T cells discussed later in chapters. Innate lymphocyte cell 2 and T helper 2 cells are type 2 immunity, and innate lymphocyte 3 and T helper 17 cell comprises type 3 immunity. In general, although innate lymphocyte cells may be active early, effector T cells rapidly become the dominant participant because they are the more numerous and make much larger amount of cytokines. The contribution of innate lymphocyte cells to host defense has been difficult to establish because it has not been possible to selectively eliminate these cells and their cytokine in experimental animals without having an impact on an analogous T cell as well. The role of innate lymphocyte cell in host defense and inflammatory disease in human is also uncertain because these cells are numerous, rare in blood, and therefore difficult to study. Natural killer cells. The natural killer cells are cytotoxic cells that play an important role in innate immune response, mainly against the viruses and intracellular bacteria. The natural killer designation derives from the fact that their major function is killing infected cells. Similar to the adaptive immune system's killer cells, the cytotoxic T lymphocyte. But unlike naive CD8 T cells, the natural killer cells are functionally competent to kill other cells when they are present in the blood or tissue without further differentiation, hence natural. Natural killer cells also secrete interferon gamma and thus resemble innate lymphocyte cells. They are also developmentally related to innate lymphocyte cell, but they are not considered identical because they are defined based on cytotoxic activity, not cytokine production. Unlike innate lymphocyte cells, which are found in peripheral tissue but are rare in the blood and lymphoid organ, natural killer cell consists of 5% to 20% of lymphocytes in the blood and spleen. They are rare in other lymphoid organ in most non-lymphoid tissue, but they are numerous in the liver and placenta. Natural killer cells in the blood appear as large lymphocytes with numerous cytoplasmic granules. The natural killer cells do not express diverse, clonally distributed antigen receptor typically of the B and T cells. Rather, they use germline DNA encoded receptor, discussed later, to distinguish pathogen infected cells from healthy cells. They can be identified in the blood by expression of CD56 and the absence of T cell markers CD3. Most of human blood natural killer cells also expresses CD16 and IgG-Fc receptor that is involved in recognition of antibody-coated cells. The function of natural killer cells. The effector function of natural killer cells are to kill infected cells and produce interferon gamma, which activate macrophage to destroy phagocytosed microbes. The mechanism of natural killer cell mediated cytotoxicity is essentially the same as that of CD8 T cytotoxic lymphocyte cell, which we will describe in detail in chapter 11. Natural killer cells like cytotoxic lymphocyte cells have granules that contain protein that mediate killing of target cell. When natural killer cells are activated, granule exocytosis release this protein adjacent to the target cell. One natural killer cell granule protein called perforin facilitates the entry of other granule protein called granzyme into the cytosol of the target cell. The granzymes are proteolytic enzymes that initiate a sequence of signaling events that cause death of target cells by apoptosis. By killing cells infected by virus and intracellular bacteria, natural killer cells eliminate reservoir of infection. Early in the course of viral infection, natural killer cells are expanded and activated by recognition of activating ligands on the infected cell and by the cytokines interleukin 12 and 15, and they kill infected cell before antigen-specific cytotoxic T lymphocyte can become full active, which usually takes 5 to 7 days. Natural killer cells also may be important later in the course of viral infection by killing infected cells that have escaped the cytotoxic lymphocyte cells mediated immune attack by reducing expression of class 1 major histocompatibility complex MHC molecule. Some tumors, especially those of hematopoietic origin, are target of natural killer cell because the tumor cells upregulate ligands for activating natural killer cell receptor and do not express normal level or types of class 1 MHC molecule, which inhibit natural killer cell activation, discussed later. Natural killer cell derived in interferon gamma increases the capacity of macrophage to kill phagocytosis bacteria, similar to interferon gamma produced by T cell. See in chapter 10. This interferon gamma dependent natural killer cell macrophage interaction can control an infection with intracellular bacteria for several days or weeks, and thus allow time for T cell mediated immunity to develop and eradicate the infection. Interferon gamma produced by natural killer cell in the lymph node can also direct the differentiation of naive T cell into T helper 1 cells. Some human natural killer cells do not express CD16, nor are they cytotoxic, but they do produce abundant interferon. Predictably, deficiency of natural killer cells seen in rare individuals lead to increased susceptibility to infection by some viruses and intracellular bacteria. Activating and inhibiting receptor of natural killer cell. Natural killer cells distinguishes infected and stressed cells from a health cell, and natural killer cell function is regulated by a balance between signals that are generated by activating and inhibiting receptor. In general, the activating receptor recognizes ligands on infected and injured cell, and the inhibitory receptor recognizes ligand on healthy normal cells. When the natural killer cell interact with another cell, the outcome is determined by the integration of signal generated from the array of inhibitory and activating receptor that are expressed by natural killer cell, and that interact with ligands on other cell. Engagement of activating receptor stimulate the killing activity of natural killer cell, resulting in destruction of stressed or infected cell. In contrast, engagement of inhibitory receptor shut off natural killer cell activity and prevent destruction of health cell. Because of stochastic nature of the expression, there is significant diversity in the array of activating and inhibiting receptor that differentiate natural killer cell expression in any one individual. The result of this is that individual natural killer cell, even in the same person, may respond to cell infected with different types of microbes. Activating receptor on natural killer cell recognize a heterogeneous group of ligands, some of which may be expressed on the normal cell and other mainly on cells that have undergone stress and are infected with microbes or are neoplastic. Many of the natural killer cell activating receptor are called killer cell Ig ligand receptor, KIR, because they contain a structural domain named Ig fold, first identified in antibody, also known as Ig molecule, discussed in chapter 5. All protein with Ig fold are member of Ig superfamily. A second important group of activating natural killer cells are similar to the family of cis-type lectin, which are protein with carbohydrate binding properties similar to that of C-like receptor discussed earlier in the chapter, although these natural killer cells do not bind carbohydrate. One well-studied lectin-like natural killer cell activating receptor is natural killer NKG2D, which bind class 1 MHC-like protein, including MHC class 1 related gene, MYC-A and MYC-B, and the protein of ULBP family. The expression of all these NKG2D ligand is increased by cellular stress, so they are found on virally infected cell and tumor cell, but not in normal cell. The NKG2D receptor associated with a signaling subunit named as DAP10, which has signaling function that stimulates natural killer cells cytotoxic against the target cell. Another important activating receptor on natural killer cells is CD16, FcR111A, which is a low affinity receptor for IgG antibodies antibody molecules have highly variable antigen binding ends, and on the opposite end, they have an invariant portion called Fc region that interact with various other molecules in the immune system. We will describe the structure of antibodies in detail in chapter 5, but for now, it is sufficient to know that CD16 bind to the Fc region of certain types of antibodies called IgG1 and IgG3. The CD16 associate with one or two different signaling properties of FCE, RI, gamma, and delta. During an infection, the adaptive immune system produces IgG1, and IgG3 antibodies that bind to microbial antigen expressing on the surface of infected cell. And CD16 on natural killer cell can bind to the Fc region of these antibodies. As a result, CD16 generates activating signal through its association signaling partner, and natural killer cells kill the infected cell and have been coated with antibody molecules. This process is called antibody-dependent cell-mediated cytotoxic. It is an effector mechanism of adaptive immunity, which we will discuss in chapter 13 when we consider humoral immunity. Inhibitory receptor of natural killer cell recognize class 1 MHC molecule, which are cell surface protein normally expressed in all healthy nucleated cell in the body. A major function of class 1 MHC molecule, distinct from their role in regulating natural killer cell activation, is to display peptide derived from cytosolic protein, including microbial protein, on the cell surface for recognition by CD8 T lymphocytes. We'll describe the structure and function of MHC molecule in relation to the T cell antigen recognition in chapter 6. For now, it is important to understand that natural killer cell uses fundamentally different types of receptor that do T cell to recognize class 1 MHC molecule. These natural killer cell receptor for class 1 molecule inhibit natural killer cell activation. This is useful because normal cells express a class 1 MHC molecule, and many viruses and other cause of cell stress lead to loss of cell surface expression of class 1 molecule. Thus, natural killer cell interpret the presence of class 1 MHC molecule as marker of normal health cells, and their absence is an indication of infection or damage. As a result, natural killer cell will be inhibited by health cell but will not receive inhibitory signal from infected or stressed cell. At the same time, the natural killer cells also likely to receive activating signal from infected or damaged cell through activating receptor. The net result will be activation of natural killer cell to secrete cytokines and to kill the infected or stressed cell. This ability of natural killer cells to become activated by host cell that lack class 1 molecule has been called the recognition of missing self. The largest group of natural killer cell inhibitory molecule receptor belong to the same KIR family that include activating receptor discussed earlier. The inhibitory KRI bind a variety of different class 1 molecules. Other inhibitory receptors are lectin-like, such as CD94, NKG2A heterodimer, which recognize a class 1 molecules called HLA, HLAE. Interestingly, HLAE display peptide derived from other class 1 molecules. So CD94 NKG2A is surveillance receptor for several different class 1 MHC molecules. Activating an inhibitory natural killer receptor contain a structural motif in their cytoplasmic tail that engage the signaling pathway that respectively promote or inhibit target cell killing and cytokine secretion. Activating receptor have immunoreceptor tyrosine-based activation motif items. Immunoreceptor tyrosine based activation motif, which contain a tyrosine residue that become phosphorylated to cytoplasmic kinase after binding to ligand to the receptor. Other protein kinases are recruited to the modified item and become activated, and these kinases contribute to further signaling by phosphorylation additional protein, eventually leading to cytotoxic activities and the cytokine secretion. Items also found in the cytoplasmic tail of other signaling receptor in the immune system, including the antigen receptor complex T and B cell, and we will discuss their structure and signaling function in more detail in chapter 7. In some activating receptor, a single polypeptide chain contains the cytoplasmic item and a cellular ligand binding portion. In other receptor, the items are in separate polypeptide chain, such as FCERI, so named because it was first identified as a signaling chain of FCE receptor, chapter 20, a component of TCR complex, a DAP12. These signaling protein do not bind ligand but are non-covalently associated with ligand binding chains. Inhibitory receptor of natural killer cell have immunoreceptor tyrosine-based inhibition motif, which engage molecules that block the signal pathway of activating receptor. Items contain tyrosine residue that are phosphorylated when ligand bind to the inhibitory receptor and then serve a docking site of recruitment and activation of tyrosine phosphatase, which remove the phosphate from several signaling proteins generated by signaling downstream of natural killer cell activating receptor. The end result is blocking of signaling function of activating receptor. Item also found in cytoplasmic tails of other receptor beside natural killer cell inhibitor receptor, and we will discuss their structural and signaling function in more detail in chapter 7. KIR gene are polymorphic, meaning that there are several allelic variants in human population. As a result, one person may express different receptor than another person. Some group of KRI allele are often inherited together from single parent. This group of linked genes are called KRI haplotypes. There are two major KRI haplotypes and some are rare ones. Haplotypes differ in number and receptor encoded, and some have more or fewer activating receptor than others. Some haplotypes are associated with increased susceptibility to some disorder, including spontaneous abortion and a type of eye inflammation called uveitis. Cytokines can enhance a functional response of natural killer cell. The major cytokines of innate immune system that enhance natural killer cells function are interleukin 12, interleukin 15, interleukin 18, and type 1 interferons discussed later. Each of these cytokines enhance the cytotoxic activities of natural killer cell, and they can stimulate the interferon gamma secretion by natural killer cell, independent of activating receptor. In addition, interleukin 15 is important growth factor for natural killer cell. T and B lymphocyte with limited antigen receptor diversity. In contrast to the cells of innate immune system that we have discussed so far, most of T and B cell lymphocyte of adaptive immune system use highly diverse receptor to recognize an enormous variety of a different antigen. Certain small population of lymphocyte expresses antigen receptor that are structurally the same as those of T B cells, but these receptors have very little diversity. This T and B cell subset may recognize structure expressed by many different or commonly encountered microbial species. T cell with limited antigen receptor diversity include invariant natural killer cell, invariant natural killer cell, INKT cell called alpha delta T cell, mucosa associated invariant T cell, MITE, and intraepithelial T cell with alpha-beta T cell receptor mentioned earlier. B cell subsets that produce antibody with a limited set of specificities include B1 cell and marginal zone B cell. Although these T and B cells perform functions similar to those of their more clonally diverse counterparts, the nature of their specificities places them in a special category of lymphocytes that is more akin to cell of innate immunity than to cell of adaptive immunity. These special T and B cell subsets are described in chapter 10 and 12 respectively. Mast cell. Mast cell are sentinel cells present in the skin, mucosal epithelial, and connective tissue that rapidly secrete pro-inflammatory cytokines and lipid mediator in response to infection and other stimuli. We introduced mast cell in chapter 2. Recall that these cells contain abundant cytoplasmic granules filled with various inflammatory mediators that are released when the cells are activated, either by microbial products or by special antibody-dependent mechanism. The granule content include vasoactive amines such as histamine that causes the vasodilatation and increase the capillary permeability, and proteolytic enzyme that can kill bacteria or inactivate microbial toxin. Mast cell also synthesize and secrete lipid mediators such as leukotrienes and prostaglandins, and cytokines such as tumor necrosis factor. Because mast cells are usually located in the adjacent blood vessel, they are released granule content rapidly induce changes in the blood vessel that promote acute inflammation. Mast cell expresses toll-like receptors and toll-like receptor ligands that can induce mast cell degranulation. Mast cell deficiency in mice are impaired in controlling bacterial infection, probably because of defective innate immune response. Mast cell products also provide defense against helminthes and responsible for symptoms of allergic disease. We will return to a detailed discussion of the mast cell in the context of allergic disease in chapter 20. Soluble effector molecule of innate immunity. Several different kinds of molecules that recognize microbes and promote innate response exist in soluble form in the blood and extracellular fluid. These molecules provide ill defense against pathogens that enter the circulation or are present outside the host cell. At some stages of their life cycle, the soluble effector molecule function in two major ways. The soluble effector molecules function in two major ways. One, by binding to microbes, they act as opsin and enhance ability of macrophage and neutrophil to phagocytize the microbes. This is because the phagocytic cells expresses membrane receptor specific for opsin, and these receptors can efficiently mediate the internalization of the complex of opsin and bound microbes and subsequent destruction of ingested microbes. After binding to microbes, soluble mediator of innate immune promote inflammatory response that bind to more phagocytes to site of infection, and they may also directly kill the microbes. Soluble effector molecules are sometimes called a humoral branch of innate immunity, analogs to humoral branch of adaptive immunity mediated by antibodies. The major component of humoral innate immune system are complement system, collectins, pentrataxin, ficolin, which are described next. The complement system. The complement system consists of several plasma proteins that work together to opsonize microbes to promote the recruitment of phagocytes to a site of infection, and in some cases to directly kill the microbes. Complement activation involves proteolytic cascade in which an inactive protein called zymogen is altered to become an active protease that cleave and thereby induce the proteolytic activity of the next complement protein in the cascade. Enzymic cascade result in tremendous amplification of the amount of proteolytic product that are generated at each step. These products perform the effector function of complement system. The first step is inactivation of complement system is recognition of molecule on microbial surface, and this occur in three ways, each referred to as a distinct pattern of complement activation. There is a classical pathway, so-called because it was discovered first, uses a plasma protein called C1q to detect antibody bonding on the surface of microbes and other structure. Once C1q binds to the Fc portion of antibodies, two associated serine proteins called C1r and C1s become active and initiate a proteolytic cascade involved in other complement protein. The classical pathway is one of the major effector mechanisms of humoral arm of adaptive immune response, chapter 13. Innate immune system soluble protein called pentraxin, which I discussed later, can also bind to C1q and initiate the classical pathway. The second is alternative pathway, which was discovered later but is phylogenetically older than the classical pathway, is triggered when the complement protein called C3 directly recognizes certain microbial surface structure, such as bacteria LPS. C3 is also constitutively activated at low level in blood and extravascular fluid and binds to cell surface, but it is then inhibited by regulatory molecule present in mammalian cell. Because microbes lack these regulatory proteins, the spontaneous activation can be amplified on microbial surface. Thus, this pathway can distinguish normal cell from foreign microbes on the basis of the presence or absence of regulatory proteins. The third one is lectin pathway. This is triggered by a plasma protein called mannose binding lectin, MBL, mannose binding lectin, which recognizes terminal mannose residue on microbial glycoprotein and glycolipid, similar to the mannose receptor on the phagocyte described earlier. The mannose binding lectins is a member of the colectin family discussed later, with hexameric structure similar to that of C1q component of complement system. After MBL, mannose binding lectin bind to the microbes two zymogens called MASP1, the man-nose-associated serine protease 1, or mannan binding lectin associated serine protease, and MASP-P, with functional similar to those of C1r and C1s, associate with the mannose binding lectin and initiate a downstream proteolytic step identical to classical pathway. Recognition of microbe by Na3 complement pathway results in sequential recruitment and the assembly of additional complementary protein in protein complex. One of these complexes, called C3 convertase, cleave the central protein of complement system, T3 producing C3a and T3b. The larger C3b fragment become covalently attached to the microbial surface where the complement pathway was activated. The sequential enzymatic activity of complement protein provides such a tremendous amplification of T3b molecule can deposit on surface of microbes within 2 to 3 minutes. T3b serves as opson to promote phagocytosis of the microbes. The small fragment C3a is released and stimulates inflammation by acting as chemoattractant for neutrophil by inducing mast cell degranulation and by directly increasing vesicular permeability so that plasma protein and fluid leak into site of infection. T3b bind other complement protein to form proteins called C5 converters that cleave C5, generating a released peptide of C5a and large fragment of C5b that remain attached to the microbial cell surface membrane. C5a exit the same pro-inflammatory effectors excite the same pro-inflammatory effect as T3A and is more potent. C5b initiates the formation of complex of complementary protein C6, C7, C8, and C9, which are assembled into membrane pore called the membrane attack complex, MAC, that causes lysis of the cell where complement is activated. The complement system activated by the alternative and lectin pathway is an essential component of innate immunity, and patients with deficiency in C3 are highly suspected to the recurrent, often lethal bacterial infection. Genetic deficient in MAC formation, the terminal product of classical pathway, causes increasing susceptibility to only a limited number of microbes, notably Neisseria bacteria, which have thin cell wall that make them especially susceptible to the lytic action of MAC. The complement system also contributes to the cell and tissue injury in many inflammatory and autoimmune disease. We will discuss the complement system in more detail in chapter 13. Pentraxin. Several plasma proteins that recognize microbial structure and participate in innate immune belong to the pentraxin family, which is phylogenetically old group of structural homologous pentameric protein. Prominent members of this family include shorty pentraxin, C-reactive protein, CRP, and serum amyloid, P, SAP, and the long pentraxin, PTX. Both CRP, C-reactive protein, and SAP, serum amyloid P, bind to several different species of bacteria and fungi. The molecular ligands recognize the serum reactive C-reactive proteins and serum amyloid P include phosphorylchlorine and phosphatidylethylamine, respectively, which are found on bacterial membrane and become exposed on apoptotic cells. CRP, SAP, or serum amyloid P, and PTX3 all activate complement by binding to C1q and initiate the classical pathway. Plasma concentration of CRP are very low in healthy individuals, but can increase up to 1,000-fold during infection and in response to other inflammatory stimuli. The increased CRP level results from increased synthesis by liver cell induced by cytokine interleukin-6, 1, and tumor necrosis factor, which are produced by phagocytes and dendritic cell as part of innate immune response to infection or injury. Liver synthesize and plasma level of several other proteins, including SAP and some unrelated to pentraxin, also increase in response to interleukin-1, interleukin-6, and tumor necrosis factor. All of these plasma proteins are called acute phase protein because they are elevated in the blood during acute inflammatory reaction, and their increasing production is part of the acute phase response to infection and other results. PTX3 is produced by several cell types, including the dendritic cell, macrophage, and endothelial cells, in response to toll-like receptor ligands and inflammatory cytokines, such as tumor necrosis factor, and may be considered as an acute reactant. PTX3 is also stored in neutrophil granules and released as neutrophil dyes. PTX3 recognizes various molecules and on fungal, certain bacteria and virus, as well as apoptotic cell, and activate the classical complement pathway. Studies with the knockout mice reveal that PTX3 provide protection against these microbes, including the fungus Aspergillus fumigatus and influenza virus. Colectin and ficolins. Colectins are a family of trimeric and hexameric or hexameric protein, each subunit of which contain a collagen-like tail connected by neck region to calcium-dependent C-type lectin head. 3 members of this family were soluble effector molecules in the innate immune system. These are mannose binding lectins and pulmonary surfactant protein SPA-SPD. Mannose binding lectin, which is a soluble pattern recognition receptor that binds carbohydrate with terminal mannose and fucose, were discussed earlier in relation to the lectin pathway of complement activation. Mannose binding also functions as an opson by binding to and enhances phagocytosis of microbes. Recall that opsins simultaneously bind microbes and a surface receptor on the phagocyte membrane. And in case of a mannose binding receptor, lectin, the surface receptor is called C1q receptor because it also binds to C1q. This receptor mediates an internalization of microbes that are opsonized by a mannose binding lectin. The gene encoding mannose binding lectin is polymorphic, and certain allele are associated with impaired hexamer formation and reduced blood level. Low mannose binding level, lectin level, result in increased susceptibility to a variety of infection, especially in individuals who have other immune defect. Surfactant protein A, SPA, and surfactant protein D, SPD, are collecting with lipophilic properties shared by other surfactants. They are found in alveoli of the lung, and their major function are to maintain ability of alveoli to expand upon inhalation by reducing surface tension of alveolar fluid, and as mediator of innate immune response in the lung. They bind to various microorganisms, and they act as opsin, facilitating ingestion by alveolar macrophage. Surface protein A and surface protein D can also directly inhibit bacterial growth, and they may activate macrophage. Surface protein A and surface protein D deficiency in mice has impaired ability to resist a variety of pulmonary infection. Ficolin are plasma proteins that are structurally similar to colectin. They possess a collagen-like domain, but instead of C-type lectin domain, they have fibrinogen-type carbohydrate recognition domain. Ficolins have been shown to bind several species of bacteria, opsonizing them and activating complement in a manner similar to that of mannose binding lectin. The ligands of ficolin include N-acetylglucosamine and the lipoic acid component of the cell wall of gram-positive bacteria. Now that we have discussed the general properties of various components of innate immune system, including the cellular pathogen recognition receptor, and soluble effector molecule, we can consider how these various components work to protect against pathogen. The three major mechanisms by which the innate immune system protects against infections are by inducing inflammation, inducing antiviral response, and stimulating adaptive immunity. The inflammatory response. The principal way by which the innate immune system deals with the infection and tissue injury is to stimulate acute inflammation, which is the accumulation of leukocyte, plasma proteins, and fluid derived from the blood at an extravascular tissue site of infection or injury. The leukocytes and plasma proteins, which are critical for innate defense against microbes, normally circulate in the blood and must be recruited to extravascular sites of infection or injury, where they perform the effector function that kill microbes and begin repair damaged tissue. Acute inflammatory response begins with recognition of microbial bumps or dumps from injured host cell by tissue sentinel cells, mainly macrophage, dendritic cell, and mast cell. These sentinel respond by secreting the mediator that act on small blood vessels in the way that promote increased blood flow, delivery of plasma proteins, and migration of leukocytes into the tissue. Detailed description of various mediator and pathological manifestation of inflammation can be found in pathology textbook. We will focus our discussion on particular aspects of inflammatory process that have broad relevance to both innate and adaptive immunity and immune-mediated inflammatory diseases, beginning with the description of important cytokine in innate immune inflammatory response. The major inflammatory cytokine of innate immunity. One of the earliest response of the innate immune system to infection and tissue damage is secretion of cytokine by tissue cell, which is critical for acute inflammatory response. The cytokine of innate immune have some important general properties and function. They are produced mainly by tissue macrophage and dendritic cell, although other cell types, including mast cell, endothelial cell, and some epithelial cell, can produce them. Most of these cytokines act on the cell close to their cell or of origin, paracrine action. In some severe infection, enough of cytokine may be produced so that a significant amount enter the circulation and act in distance endocrine action. Different cytokines have similar or overlapping action or are functionally unique. One cytokine may stimulate the production of other, thus setting up a cascade that amplifies the reaction or induce the new reaction. The cytokine of innate immune serves several roles, inducing the inflammation, inhibiting viral replication, promoting T cell response, limiting the innate immune response. These functions are described next and later in the chapter. Many cytokines that are produced by innate immune cells, such as tumor necrosis factor, interleukin-17, interleukin-5, interferon alpha, interferon gamma, are also produced by T lymphocyte in adaptive immune response. Three of the most important pro-inflammatory cytokines of innate immune system are tumor necrosis factor, interleukin-1, both of which we have mentioned several times, and interleukin-6. We will discuss the major features of these cytokines, focusing mainly on tumor necrosis factor, interleukin-1, before describing their role in acute inflammation. Tumor necrosis factor. Tumor necrosis factor is a mediator of acute inflammatory response to bacteria and other infectious microbes. The name of this cytokine derived from its original identification as a serum substance factor that caused by necrosis of tumor, now known as to be result of inflammation of thrombosis of tumor blood vessel. Tumor necrosis factor is also called tumor necrosis alpha, distinguish it from the closely related tumor necrosis beta, which is also called lymphotoxin. Tumor necrosis factor is produced mainly by macrophage and also by other cells types, including the dendritic cell and mast cells. In macrophages, it is synthesized as non-glycosylated type 2 membrane homotrimetric protein that is able to bind to one form of tumor necrosis factor receptor. The membrane form of tumor necrosis factor is cleaved by a membrane-associated metalloproteinase released a polypeptide fragment, and 3 of these polypeptide chains polymerize to form a triangular pyramid-shaped circulating tumor necrosis factor protein. These receptor binding sites are at the bases of the pyramid, allowing the simultaneous binding of the cytokine to 3 receptor molecules. Tumor necrosis factor is a member of large family of homologous protein called tumor necrosis factor superfamily, all of which have features of forming homotrimers. There are two distinct tumor necrosis receptor called type 1, tumor necrosis factor type 1, and type 2, tumor necrosis factor type 2. The affinity of tumor necrosis factor for receptor are usually low for cytokine, and the KD being only approximately 1 × 10⁻⁹ M for binding to tumor necrosis factor receptor 1, and approximately 5 × 10⁻⁻10 M for binding to tumor necrosis factor receptor 2. Both tumor necrosis factor receptor are present on the mast cell. These tumor necrosis factor receptor are a member of the large family of protein called the tumor necrosis factor receptor superfamily, many of which are involved in immune and inflammatory response. These receptors exist as trimer in the plasma membrane, a ligand binding to some tumor necrosis factor receptor family member, such as tumor necrosis factor receptor 1, tumor necrosis factor receptor 2, and CD40 lead to the recruitment of protein called tumor necrosis factor receptor associated factor, TRAF, to the cytoplasmic domain of receptor. The TRAF, or tumor necrosis factor receptor associated factor, activate transcription factor, notably NF kappa beta and API, seen in chapter 7, cytokine binding to some family members such as tumor necrosis factor receptor 1 may lead to the recruitment of an adapter protein that activate caspase and trigger apoptosis. Thus, different members of tumor necrosis factor receptor family can induce gene expression or cell death, and some can do both. Tumor necrosis factor production by macrophage is stimulated by pump and dump, toll-like receptor, node-like receptor, lectin-like receptor, and C-lectin D-like receptor can all include tumor necrosis factor gene expression, in part by activating the NF kappa beta transcription factor. Many different microbial products can therefore induce tumor necrosis factor production. Tumor necrosis factor has multiple local and systemic effects that account for many of the reactions in inflammation. Tumor necrosis factor is a major contributor of inflammation in several chronic inflammation disease, and anti-tumor necrosis factor agents have become mainstay of treatment for many of these diseases. Large amount of these cytokines may be produced during infection with gram-negative and gram-positive bacteria, which expresses and releases a cell wall toll-like receptor ligand, LPS, and lipotetoic acid, respectively. Septic shock, a life-threatening condition resulting from severe infection, is mediated in large part by tumor necrosis factor. We will discuss the septic shock later in this chapter. Interleukin-1. Interleukin-1 is also a mediator of an acute inflammatory response and has many actions similar to those of tumor necrosis factor. A major cellular source of interleukin-1, like that of tumor necrosis factor, is activated mononuclear phagocyte. Interleukin-1 is also produced by many cell types other than macrophage, such as neutrophil, dendritic cell, epithelial cell, that is keratinocytes, and endothelial cells. There are two forms of interleukin-1 called interleukin-1 alpha and interleukin-1 beta that are less than 30% homologous to each other, but they bind to the same cell surface receptor and have same biological activities. The main biologically active secretory form in the setting of infection and most immune response is alpha beta. Interleukin-1 production usually requires two distinct signals. One that activates new gene transcription and production of a 33 kilodalton precursor pro-interleukin-1 beta polypeptide, and second that activates the inflammasome to proteolytically cleave the precursor to generate the 17 kilodalton mature interleukin-1 beta protein. As discussed earlier in this chapter, interleukin-1 beta gene transcription is induced by toll-like receptor, node-like receptor, RL receptor signaling pathway that activate the NF kappa beta, whereas pro-interleukin-1 beta cleaves mediated by caspase 1, which is activated by inflammasome. Tumor necrosis factor can also stimulate phagocytes and other cell types to produce interleukin-1. This is an example of cascade of cytokines that have similar biological activities. Unlike most secretory protein, neither interleukin-1 alpha nor interleukin-1 beta has hydrophobic signal sequence to target the nascent polypeptide to the endoplasmic reticulum membrane. As discussed earlier, interleukin-1 beta may be secreted through a membrane pore formed by gasdermin D. Interleukin-1 mediates its biological effect through a membrane receptor called type 1 interleukin-1 receptor, which is expressed on many cell types, including endothelial cells, epithelial cells, and leukocytes. This receptor is an integral membrane protein that contains an extracellular ligand binding Ig domain and TAR signaling domain in the cytosolic region, which we described earlier in reference to toll-like receptor. The signaling event that occur when interleukin-1 bind to the type 1 interleukin-1 receptor are similar to those triggered by toll-like receptor and result in activation of NF, kappa beta, and up activation protein 1 transcription factor. A second interleukin-1 receptor called type 2 interleukin-1 receptor appear incapable of activating downstream signals and serves as a decoy receptor that limits response to interleukin-1. Interleukin-6. Interleukin-6 is another important cytokine in acute inflammatory response that has both local and systemic effects. It induces the synthesis of acute phase reactants by the liver and promotes differentiation of interleukin-17 producing helper T cell. Interleukin-6 synthesized by mononuclear phagocyte, dendritic cell, vesicular endothelial cells, fibroblast, and other cells. In response to pump and dumps, and in response to interleukin-1 and tumor necrosis factor. Interleukin-6 is homodimer that belong to the type 1 cytokine family. The receptor for interleukin-6 consists of cytokine binding polypeptide chain and a signal transducing subunit called GD130 that is also signaling component of receptor for other cytokines. The GP130 subunit is widely expressed on the many cell types, but the interleukin-6 binding chain is the only expressed by leukocyte and hepatocyte as transmembrane protein in association with GP30. However, a soluble form of interleukin-6 binding chain is generated by proteolytic cleavage of membrane form and is present in blood and tissue fluid. This soluble form can bind to interleukin-6 and then complex can associate with extracellular part of GP130 on many cell types and initiate signaling. This mechanism is called trans-signaling. The interleukin-6 receptor engage a signaling pathway that activates the transcription factor STAT3. Interleukin-6 is the major contributor to inflammation in several human inflammatory diseases, including rheumatoid arthritis, and antibodies specific for interleukin-6 receptor are used to treat some form of arthritis. Some lymphoproliferative disorders, such as Castleman disease, are caused by human herpes virus 8, a virus that encodes a homologue of interleukin-6, and interleukin-6 blockage has been also used to treat these diseases. Other cytokine producing during innate immune response. In addition to tumor necrosis factor, interleukin-1 and interleukin-6, dendritic cell and macrophage activated by pump and dump produce other cytokines that have important roles in the innate immune response. We will discuss the main features of some of these cytokines and their role in the innate immune in this section. Interferon and inhibitory cytokine are discussed later in the chapter. Interleukin-12 is secreted by dendritic cell and macrophage and stimulate interferon gamma production by interleukin by innate lymphocyte cells, natural killer cell, and T cell, enhances natural killer cell and cytotoxic T cell lymphocyte mediated cytotoxicity, and promote differentiation of T helper 1 cell. Interleukin-12 exists as disulfide-line heterodimer of 35 kilodalton P35 and 40 kilodalton P40 subunit. The P35 subunit is the member of the type 1 cytokine family, and the P40 subunit is also a component of cytokine interleukin-23, which is involved in differentiation of T helper 17 cells. Therefore, an antibody specific for P40 blocks both interleukin-12 and interleukin-23, and thus inhibit the interleukin-12 dependent development of T helper cell and the interleukin-23 dependent development of T helper 17. This antibody is approved for the treatment of inflammatory bowel disease and psoriasis, which are caused by T helper 1, T helper 17 cytokines. The principal source of interleukin 12 are activated dendritic cell and macrophage. Many cells appear to synthesize the P35 subunity but macrophage and dendritic cell are the main cell that produce the P40 component and therefore biologically active cytokine. During innate immune reaction to microbes, interleukin 12 is produced in response to toll-like receptor and other pattern recognition receptor signaling induced by many microbial stimuli, including bacterial LPS or lipoteichoic acid and virus infection. Interferon gamma produced by natural killer cell and T cell also stimulate interleukin 12 production, contributing to a positive feedback loop. The receptor for interleukin 12 is heterodimer composed of B1 and B2, beta 1 and beta 2 subunit, both of which are a member of the type 1 cytokine receptor family. Both chains are required for high affinity binding of interleukin 12 and signaling, which activate the transcription factor STAT4 expression of beta 2 chain of the interleukin 12 receptor is itself enhanced by interferon gamma, whose production is stimulated by interleukin 12. This is an example of positive amplification in the loop in the immune response. Studies with the gene knockout mice and the phenotype of rare patient with the mutation in the interleukin 12 receptor support the conclusion that interleukin 12 is an important interferon gamma production by natural killer cell and T cell and for the host resistance to the intracellular bacteria and some viruses. For example, patients with the mutation in interleukin 12 receptor beta 1 subunit have been described and they are highly susceptible to infection with intracellular bacteria, notably Salmonella and Mycobacteria, Interleukin 12 secreted by the dendritic cell during antigen presentation to naive CD4 T cell promote differentiation of this cell into T helper 1 subset of helper T cell, which are important for defense against intracellular infection. This is the key way in which innate immune shape adaptive immune response. Interleukin 18 enhances the function of natural killer cell, similar to interleukin 12. Recall that the production of interleukin 18, like the interleukin 1, is dependent on the inflammasome. Also, like interleukin 1, interleukin 18 binds to the receptor that signals through a TER domain. Children with gain of function mutation in NLRSC4 have very high level of inflammasome generated interleukin 18 produced by intestinal epithelial cell and suffer from systemic macrophage activation syndrome, likely due to excessive interleukin 18 driven production of interferon gamma from natural killer cell. Interleukin 15 stimulates growth of and function of innate lymphocyte cells, natural killer cell, and some T cells. Interleukin 15 is structurally homologous to the T cell growth factor, interleukin 2, and the heterodimeric interleukin 15 receptor share 2 subunity with the interleukin 2 receptor. An interesting feature of interleukin 15 is that it can be expressed on the cell surface bound to the alpha chain of its receptor, and in this form can be presented to and stimulate nearby cell that expresses the receptor composed of the other 2 chains, beta and gamma. Interleukin 15 presented this way by dendritic cell to natural killer cell in lymph nodes activates signaling pathway that promote natural killer cell interferon gamma production. Interleukin 15 also serves as survival factor for natural killer cell and memory CD8 T cells. Interleukin 25 thermic stroma lymphopoietin TSLP and interleukin 33 are structurally unrelated cytokine produced by epithelial barrier cells as well as other cell types, which stimulate the interstitial lymphocyte cells, T helper 2 cells, and mast cell to produce interleukin-4, interleukin-5, and interleukin-13. The later cytokines are important for defense against helminthes, but also contribute to the allergic disease. Interleukin-33 is constitutively expressed by barrier epithelial cell and stored in the nucleus. It is often called alarmin because it is rapidly released from the damaged epithelial cells and stimulates the innate and adaptive immune response. In addition to cytokine discussed here, other cytokines play important roles in both innate and adaptive immune response, including interleukin-5, interleukin-17, interferon gamma, interferon gamma. This cytokine will be discussed in detail in chapter 10. Then, when we consider the helper T cell subset that produce them. Sequence of event in inflammation, vascular changes, and leukocyte migration into tissue. Acute inflammatory response are initiated when a sentinel cell, including the mast cell, tissue-resident macrophage, and dendritic cell, which are present in normal tissue before infection, use the toll-like receptor and the cytosolic innate pattern recognition receptor to sense microbes and injured cells. Mast cells respond to bump and dumps by secreting histamine and prostaglandin that cause vasodilatation and increase capillary permeability, which increases blood flow through the tissue and increase movement of the plasma protein such as complement protein, pentraxin, colectin, and antibodies about the blood vessels. Antibodies out of the blood vessels. These soluble innate effector molecules work in tandem with the leukotriene that are recruited into the tissue from the circulation. Recruitment of the large number of neutrophils followed by monocytes from the blood into tissue typically occur as part of the acute inflammatory response to infection and tissue injury. The vasodilatation caused by mast cells-derived histamine and prostaglandin increases the number of these leukocytes entering affected tissue and also changes blood flow characteristic that enhances typical physical interaction of circulating leukocyte with the blood vessel wall. Tumor necrosis factor, interleukin-1, 6, and chemokines, all of which are produced by sentinel cell, a site of infection or tissue injury, have multiple effects on capillaries, venules, leukocyte, and bone marrow, which together increases the local delivery of cells that can fight infection and repair tissue. Leukocyte recruitment was described in chapter 3 and will be considered only briefly here. Postcapillary venule endothelial cell increases surface expression of adhesion molecule for leukocyte, expression of E-selectin and ligands for integrin, including intercellular adhesion molecule 1, ICAM1, and vesicular cell adhesion molecule 1, VCAM1, are induced by tumor necrosis factor and interleukin-1 activation of a transcription factor, including the NF-kappa beta. P-selectin stored in the endothelial cell cytoplasmic granule may be mobilized to cell surface in response to thrombin generated by coagulation cascade. Tumor necrosis factor and interleukin-1 also stimulate various cells to secrete chemokines, and such as CXCL8 and CCL2, which bind to the receptor on neutrophil and monocyte respectively. As discussed in chapter 3, these chemokines increase the affinity of leukocyte integrin for their ligands, stimulating the directional movement of leukocyte. The result of increased selectin, integrins, and chemokines expression is an increase in neutrophil and monocyte adhesion to endothelial cells and transmigration through the vessel wall. The leukocyte accumulate in the tissue, forming an inflammatory infiltrate. The action of tumor necrosis factor on endothelial and leukocyte are critical for local inflammatory response to microbes. If inadequate quantities of tumor necrosis factor are present, e.g. in a patient treated with a drug that block tumor necrosis factor or in tumor necrosis factor gene knockout in mice, a consequence may be failure to contain infection. In addition, Tumor necrosis factor, interleukin-1 and interleukin-6 produced by inflammatory sites may enter the blood and be delivered to the bone marrow, where they work in concert with the colony-stimulating factor to enhance the production of neutrophil from bone marrow progenitor and release the mature neutrophil in the blood. In this way, this cytokine increase the supply of cells that can be recruited to the site of infection and replace leukocytes that are consumed during inflammatory reaction. Typically, the leukocyte that is recruiting first into the site of inflammation is the neutrophil because typically the leukocyte that is recruited first into the site of inflammation is neutrophil because it is the most abundant leukocyte in the blood and the most rapid responder to chemoattractant signal. Blood monocytes, which become macrophage in the tissue, become increasingly prominent over time and may be dominant population in some reaction. Neutrophil and monocyte-derived macrophage phagocyte and phagocytose and kill microbes, discussed later, and these functions are enhanced by opsonization of microbes by complementary protein, antibodies, and other soluble mediators of an innate immunity. Furthermore, the recruitment of phagocytes, especially the macrophage, respond to the microbes and injured cell by secreting more inflammatory cytokine and chemokines, which promote more leukocyte recruitment and amplify the acute inflammatory response. Acute inflammations can develop in minutes to hours and last for days. Chronic inflammation is the process that takes place from acute inflammation if the infection is not eliminated or tissue injury is prolonged. It is usually involve recruitment and activation of monocyte and lymphocyte. Chronic inflammatory sites also often undergo tissue remodeling with angiogenesis and fibrosis. Although innate immune stimuli contribute to chronic inflammation, the adaptive immune system is usually also involved, and cytokines produced by T cell are powerful inducers of chronic inflammation. Engagement and killing of microbes by activated phagocytes. Neutrophil and macrophage that are recruited into the site of infection ingest microbes into vesicle by process of phagocytosis and destroy these microbes. Phagocytosis is an active, energy-dependent process of engulfing the large particle greater than 0.1 micrometer in diameter into vesicle. Phagocytic vesicle fuse with lysosomes and the ingested particle are destroyed. In this way, the mechanism of killing, which could potentially injure the phagocyte, isolated from the rest of the cell. Neutrophil and macrophages expresses receptor that specifically recognize microbes, and binding of microbes to these receptor is the first step in phagocytosis. Some of these receptors are pattern recognition receptor, including C-type lectin and scavenger receptor, which we discussed earlier. The pattern recognition receptor can contribute to the phagocytosis only of organisms that expresses particular molecular patterns, such as ammonose for ammonose receptor. Phagocytes also have high affinity receptor for certain opsins, including antibody molecule, complement protein, and protein lectin. These receptors are critical for phagocytosis of many different microbes that are coated with opsin. Coating microbes with antibodies is one of the most efficient system of opsonization. Phagocyte expresses a high affinity Fc receptor called Fc specific for one type of antibody called IgG, seen in chapter 5 and chapter 13. Thus, if an individual responds to an infection by making IgG antibody against microbial antigens, the IgG molecule binds to these antigens. The Fc ends of the bound antibody can interact with Fc gamma R1 on phagocytes, and the end result is efficient phagocytosis of the microbes. Antibody-dependent phagocytosis illustrates a link between innate and adaptive immunity antibodies. Innate and adaptive immunity. Antibodies are product of adaptive immune system, B lymphocyte, that engage in innate immune system effect cell phagocyte to perform their protective function. Once a microbes or particle bind to receptor on phagocyte, the plasma membrane in the region of the receptor begin to invaginate and extend a cup-shaped projection around the microbes. When the protruding membrane cap extends beyond the diameter of the particle, the top of the cap closes over and pitches off the interior of the cap to form an inside-out intracellular vesicle. This vesicle, called a phagosome, containing ingested foreign particle, and it breaks away from the plasma membrane. The cell surface receptor also deliver activating signal that stimulate microcidal activity of phagocytes. Phagocytosis, the microbes are destroyed as described next, and at the same time peptides are generated from microbial protein and presented to T lymphocyte to initiate adaptive immune response. Activated neutrophil and macrophage kill phagocytes microbes by action of a microcidal molecule in phagolysosomes. Signal from various receptors, including pattern recognition receptor, such as toll-like receptor, opsin receptor, such as FC and CB receptor, the receptor of cytokines, mainly interferon gamma, and CD4 function cooperatively to activate phagocytes to kill ingested microbes. Fusion of phagocytic vacuoles, which lysosome result in the formation of phagolysosome, where most of the microcidal mechanisms are concentrated. Three classes of microcidal molecules are known to be the most important. There is a reactive oxygen species, ROS, activated neutrophil, and to a lesser extent macrophage convert molecule oxygen into reactive oxygen species, which are highly reactive oxidized agents with a free radical that destroy microbes and other cells. The primary free radical generating system is phagocytes oxidase system. The phagocyte oxidase is a multi-subunit enzyme that is assembled in activated phagocytes, mainly in the phagolysosome membrane. Phagocyte oxidase is activated by many stimuli, including interferon gamma and signal from toll-like receptor. The function of this enzyme is to reduce the molecular oxygen into ROS, reactive oxygen species, such as superoxide radical with the reduced form of nicotinamide adenine dinucleotide phosphate, acting as cofactor. Superoxide is enzymically dismutated into hydrogen peroxide, which is used for enzyme myeloperoxidase to convert normally and reactive halide ions into reactive hypohalous acids that are toxic for bacteria. This process by which a reactive oxygen species are produced is called a respiratory burst because it requires oxygen consumption, cellular respiration. As disease called the chronic granulomatosis disease is caused by inherited deficiency of one of the component of phagocytosis, as does this deficiency compromise the capacity of phagocytes to kill certain species bacteria. Nitric oxide. Macrophage produce a reactive nitrogen species, mainly nitric oxide, by the action of enzyme called inducible nitric oxide synthase. Inducible nitric oxide synthase is a cytosolic enzyme that is absent in resting macrophage but can be induced in response to microcidal product that activate toll-like receptor, especially in combination with interferon gamma. Induced nitric oxide synthetase catalyze the conversion of adenine to citrulline and free diffusible nitrogen gas is released. With phagolysosomes, nitrogen oxide may combine with hydrogen peroxide or superoxide generated by phagocytes oxidase to produce highly reactive peroxynitrate radical that can kill microbes. The cooperative and the reductant function of reactive oxygen species and nitric oxide is demonstrated by finding that the knockout mice lacking both induced nitrogen oxide synthetase and phagocyte oxidase are more susceptible to bacterial infection than single phagosome in oxidase or inducible nitric oxide as synthetase knockout animal. Proteolytic enzyme. Activated neutrophil and macrophage produce several proteolytic enzymes in the phagolysosomes that function to destroy microbes. One of the important enzymes in neutrophil is elastase, a broad-spectrum serine protein known to be required for killing types of bacteria. Another important enzyme is cathepsin. G-Mouse gene knockout studies have confirmed the essential requirement for this enzyme in phagocyte-killing bacteria. Microbe within macrophage may also be killed when macrophage undergo inflammasome-mediated pyrotosis, as described earlier. Neutrophil also killed microbes by extruding their DNA and granules content, which form extracellular thread on which bacteria and fungi are trapped and killed. The extruded chromatin content, which are called the neutrophil extracellular traps, nets, are composed of standard of DNA and histones to which high concentration of antimicrobial granules content are bound, including lysosomes, elastase, and defensin. Net formation require acetylation of histone by enzyme peptidyladenine deaminase, PAD4, as well as neutrophil serine proteins elastase, myeloperoxidase, and phagocytes oxidase. The extrusion of the nuclear content during net formation lead to the neutrophil cell death, referred to as netosis. The importance of net in innate protein protection against infection remain unclear, but growing evidence indicate that excessive net formation contributes to autoimmune and other inflammatory diseases. Role of macrophage in tissue repair. Acute inflammation is often associated with a significant death of host cell, including tissue cell killed by microbes from collateral damage by microcidal activities of recruited leukocytes. Once the offending agents are eliminated, the damaged tissue has to be repaired. Macrophages play a critical role in the repair process because of several activities. Macrophages clear dead cells and secrete growth factor that promote generation and angiogenesis. Macrophages also secrete tumor growth factor, beta, and other cytokines that stimulate collagen synthesis or by fibroblasts, thus promoting the formation of scar tissue to replace the damaged parts. Macrophages activated in different ways are responsible for different functions. Classically, activated macrophages are microbicidal and promote inflammation early in the reaction, and the alternative activated macrophages promote tissue repair. These pathways of macrophage activation are discussed in more detail in Chapter 10. Systemic in the pathological consequences of inflammation. Tumor necrosis factor, interleukin 1 and 6, produced during the innate immune response to infection or tissue damage, have systemic effects that contribute to the host defense and are responsible for many clinical manifestations of infection and inflammation disease. Fever. Tumor necrosis factor and interleukin 1 act on hypothalamus to induce increase in body temperature, fever. These cytokines are therefore called endogenous pyrogens. This is the host-derived fever-causing agent to distinguish them from LPS, which were considered as exogenous microbe-derived pyrogen. This distinction is mainly historically significant because we now know that even LPS induce fever by stimulating production of cytokine, tumor necrosis factor, and interleukin 1. This cytokine increases synthesis of prostaglandin in hypothalamic cells, and prostaglandin stimulate the production of neurotransmitter that rests on the body steady-state temperature to higher level by reducing heat loss. With vasoconstriction and increased heat generation through effect of skeletal and fat, prostaglandin synthesis inhibitor such as aspirin reduces fever by blocking this action of cytokines. The role of fever in host defense is not well understood, but may relate to the enhanced metabolic function of immune cells, impaired metabolic function of microbes, and change in behavior of febrile hosts that reduces risk of worsening infection and injury. Leukocytosis. Tumor necrosis factor, interleukin 1 and interleukin 6 produced at inflammatory sites circulate to bone marrow and promote the release of neutrophil and monocyte and other cytokine called colony-stimulating factor. Stimulate the production of this cell, leading to elevated number of white cells in the blood, leukocytosis. With this enhanced leukocyte migration to tissue, which can aid in antimicrobial defense, but also contribute to the inflammatory damage to tissue. Acute phase response. Tumor necrosis factor interleukin 1-6 induce hepatocyte to produce acute phase protein including CRP, SAP, and fibrinogen, which are secreted into the blood. Elevated plasma level of these proteins are commonly used clinically as sign of infection or other inflammatory process. The pentraxin CRP and SAP sub play a protective role in infection, as well as we discussed earlier in the chapter. The fibrinogen, the precursor of fibrin, contribute to the homeostasis of tissue repair. In severe infection, tumor necrosis factor may be produced in large amount and causes systemic, clinical, and pathological abnormality. If stimulus for cytokine production is sufficiently strong, large amount of tumor necrosis factor may be made into the bloodstream and act as distant sites. The principal pathological action of tumor necrosis factor as follows. Tumor necrosis factor inhibits myocardial contractility and vesicular smooth muscle tone, resulting in marked decrease in blood pressure. A shock. Inhibiting myocardial contractility and vesicular smooth muscle tone. Tumor necrosis factor causes intravascular thrombosis, mainly as a result of impairment of normal anticoagulant properties of endothelium. Tumor necrosis factor stimulates endothelial cell expression of tissue factor, a potent activator of coagulation, and inhibits expression of thrombomodulin, an inhibitor of coagulation. And endothelial alteration are exacerbated by activation of neutrophil, leading to vesicular plugging of the cell. Prolonged production of tumor necrosis factor cause wasting of muscle and fat cell, called cachexia. This wasting results from tumor necrosis factor-induced appetite suppression and reduced synthesis of lipoprotein lipase, and enzyme needed to release fat acid from circulating lipoprotein so that they can be used by tissue. A systemic complication of severe infection, usually bacterial or fungal, is a syndrome called sepsis, clinically characterized by fever, fast heart rate, and respiratory rate, metabolic abnormalities, and mental disturbance. The infection may involve microbes in the blood, but this is not documented in most cases. Bacterial sepsis is the most often initiated by LPS, also called endotoxin, released from gram-negative bacteria, or lipotetoic acid released from gram-positive bacteria, which may enter the bloodstream. Toll-like receptor signaling is then induced in cell in many group by LPS or lipoprotein acid, leading to the production of tumor necrosis factor and other cytokines, including interleukin 12, interferon gamma, interleukin 6, and interleukin 1. In the most severe form of sepsis, called septic shock, there is a vesicular collapse and disseminated intravascular coagulation caused by effect of high doses of tumor necrosis factor discussed earlier. The concentration of serum tumor necrosis factor may be predictive of outcome of severe sepsis. Septic shock can be reproduced in experimental animal by administration of LPS, lipoprotein acid, and tumor necrosis factor. Antagonists of tumor necrosis factor can prevent mortality in the experimental model, but clinical trial with anti-tumor necrosis factor antibody or with soluble tumor necrosis receptor have not shown benefit in patients with sepsis. The cause of this therapeutic failure is not known. but it may be because other cytokine elicit the same response as tumor necrosis factor. A syndrome similar to septic shock may occur as a complication of non-infectious disorder such as severe burn, trauma, pancreatitis, and other serious conditions. This has been called a systemic inflammatory response syndrome, SARS. Acute inflammation may cause tissue injury because of effector mechanism that leukocytes use to kill microbes are also toxin to host tissues. The proteolytic enzymes ROS, reactive oxygen species, and nitrogen oxide produced by phagocytes that accumulate at the site of infection can injure host cell and degrade extracellular matrix if they are generated in large quantities, especially if the microbes resist are being killed and contribute to the stimulation to stimulate the innate immune response. In fact, at least part of pathological process associated with infection is due to inflammatory response and not the direct toxic effect of microbes. Acute inflammation also causes tissue damage in the setting of autoimmune disease, in which case neutrophil and macrophage accumulate and become activated as a result of stimulation of adaptive immune system by self-antigen. As in inflammation induced by infection, tumor necrosis factor interleukin 1, 6, and 12 are key inducer of inflammation in autoimmune disease. Antagonists against all of these cytokines or their receptor are in clinical use to reduce inflammation in patients with inflammation disease such as rheumatoid arthritis, inflammatory bowel disease, and psoriasis. Antiviral response. The major way by which the innate immune system blocks the viral infection is to induce expression of type 1 interferons, whose most important action is to inhibit viral replication. Earlier in the chapter, we discussed how several pattern recognition receptors, including some toll-like receptor, node-like receptor, RLRs, CDS, generate a signal that stimulate interferon alpha and interferon beta gene expression in many different cell types. These type 1 interferon are secreted from cell and act on other cells to prevent spread of viral infection. This section will describe the properties of type of interferon and the antiviral action of this cytokine. The antiviral function of type 1 interferon are mimicked by type 3 interferons made up of membrane of interferon alpha family, which are the main antiviral interferon made by main epithelial by conventional dendritic cell. Type 1 interferon are a large family of structure-related cytokines that mediate the early innate immune response to viral infection. The term interferon derives from the ability of these cytokines to interfere with viral replication. There are many type 1 interferon which are structurally homologous and are encoded by genes in the single cluster of chromosome 9. The most important type of interferons in viral defense are interferon alpha, which include 13 closely related protein, and interferon beta, which is the single proteins. Plasmacytoid dendritic cell are the major source of interferon alpha, but it is also may be produced by mononuclear phagocyte. Interferon beta is produced by many cell types in response to viral infection. The most important stimuli for interferon synthesis are viral nucleic acid. Recall the RIG-like receptor and DNA sensor in the cytosol, and toll-like receptor 3, 7, 8, and 9 in the endosome vesicle recognize microbial nucleic acid and initiate signaling pathway that activate the IRF family of transcription factor, which stimulates the transcription of interferon gamma genes. The receptor for interferons which bind to the interferon alpha and interferon beta is heterodimer of two structure-related polypeptides. Interferon receptor 1 and interferon receptor 2, which are expressed on the nucleated cell, this receptor signal to activate STAT1, STAT2, and the IRF9 transcription factor, which induce the expression of several different types of those proteins products contribute to the antiviral defense in various ways. Type 1 interferons activate transcription of several genes that confer on cell a resistance to viral infection called antiviral state. Type 1 interferons induces gene include double-stranded RNA activated serum threonine protein kinase, which block viral transcriptional and translational events, and 2,5 oligoadenylate synthetases and the RNase L, which promote viral RNA degradations. The antiviral action of type 1 interferon is primary paracline action in that a viral infected cell or plasmacytoid dendritic cell activated by viral pump secrete interferon and act on and protect neighboring cells that are not yet infected. The effect of type 1 interferon are not specific to viral genes expression, and part of ability of these cytokines to block the spread of infection is due to their toxicity to host cells that are near the infected cell. Interferon secreted by an infected cell may also act in an autocrine fashion to inhibit viral replication in that cell. Tumor necrosis factor causes a sequestration of lymphocytes in lymph nodes, thus maximizing the opportunity for encounter with the microbial antigens. So the mechanism of this effect of type 1 interferon is the induction of molecule on lymphocyte called CD69, which form a complex with and reduce surface expression of sphingosine 1 phosphate. Receptor CYP1. Recall from chapter 3 that lymphocyte egress from lymphoid tissue depend on CYP1 binding to CYP1 receptor 1. Therefore, reduce this CYP1 inhibit this ingress and keeps the lymphocyte in lymphoid organs. Type 1 interferons increases the cytotoxic of natural killer cell and CD8 cytotoxic lymphocyte cell and promote the differentiation of naive T cell to T helper 1 subset of helper T cell. This effect of type 1 interferon enhance both innate and adaptive immune against intracellular infection, including viruses and some bacteria. Type 1 interferons upgrade expression of class 1 MHC molecules and thereby increases the probability that a viral infected cell will be recognized and killed by CD8 cytotoxic lymphocyte cells. Virus specific to CD8 cytotoxic lymphocyte cells recognize peptide derived from viral protein bound to class 1 molecule on surface of infected cell. We'll discuss the detail of T recognition of peptide MHC and cytotoxic lymphocyte killing cell in chapter 6. MHC synthesized by a viral infected cell type 1 interferons will increase the number of viral peptide class 1 MHC complex on the cell surface that the cytotoxic can see and respond to. The end result is increased killing virus infected cell and the eradication of viral infections. Thus, the principal activities of type 1 interferon work in concert to combat viral infection. Patients who develop severe COVID-19 disease caused by SARS-CoV-2 virus often show defect in type 1 interferon. About 10% of severely ill patients produce autoantibody against their own types, which may predate the infection, and the other 3.5 to 4 have inherited mutation that affect type 1 interferon production or signaling. Knockout mice lacking the receptor for type 1 interferons are susceptible to viral infection. Interferon alpha is in clinical use as an antiviral agent in certain form of viral hepatitis. Interferon gamma is also used for treatment of some tumor, perhaps because it boosts the cytotoxic lymphocyte activities and inhibits cell proliferation. Interferon gamma is used as therapy for multiple sclerosis by mechanism of its beneficial effect on this disorder is not known. Protection against viruses is due to in part to the activation of intrinsic apoptotic death pathway in infected cell and enhanced sensitivity to the extrinsic inducers of apoptosis. Viral protein synthesized in infected cells may misfold and their accumulation trigger an unfolded protein response that may culminate in apoptosis of infected cell if the misfolded protein accumulation cannot be corrected. In addition, viral infected cells are hypersensitive to tumor necrosis factor induced apoptosis. So abundant tumor necrosis factor is made by plasmacytoid dendritic cell and macrophage in response to viral infection. In addition to type 1 interferon, the type 1 tumor necrosis factor receptor engage both pro-inflammatory and pro-apoptosis pathway, and viral infection can shift this balance toward apoptosis. Stimulation of adaptive immunity. The innate immune response provides a signal that functions in concert with antigen to stimulate the proliferation and differentiation of antigen-specific T and B lymphocyte. Because innate immune response is provided by initial defense against microbes, providing the initial defense against microbes, it also set in motion adaptive immune response. The activation of lymphocytes requires two distinct signals, the first being antigen and the second being a molecule that are produced during innate immune response to microbes or injured cells. This idea is called the two-signal hypothesis for lymphocyte activation. The requirement for the antigen, so-called signal 1, ensure that the ensured immune response is specific. The requirement for additional stimuli triggered by innate immune reaction to microbes, signal 2, ensure that adaptive immune response are induced when there is a dangerous infection and not when lymphocytes recognize harmful antigen, including self-antigen. The molecule produced during innate immune reaction that function as second signal for T lymphocyte activation include co-stimulator for T cell, cytokines for both T cell and B cell, and complement breakdown product for B cell. We will return to the nature of second signal for lymphocyte activation in chapter 9 and 12. The second signal generated during innate immune response to different microbes not only enhance our magnitude of subsequent adaptive immune response, but also influence the nature of adaptive response. A major function of T cell mediated immunity is to activate macrophage to kill intracellular microbes and to induce robust inflammatory response so that the sufficiently large army of phagocytes is called into the site of infection. When dendritic cells or phagocytes encounter microbes, toll-like receptor and other pattern recognition receptors stimulate the secretion of cytokines and induce the antigen presenting cell to the expression of molecule called the co-stimulator, all of which promote cell activation. Thus, the innate immune response to microbes in macrophages stimulate adaptive T cell response that help the macrophage to destroy such microbes. By contrast, the many extracellular microbes that enter the blood activate the alternative complement pathway, and some of the proteolytic product of complement activating enhance the production of antibody by B lymphocytes. See chapter 11. These antibodies opsonize the microbes and thereby promote their phagocytosis by neutrophil and macrophage, or kill the microbes by complement-dependent mechanism. Thus, blood-borne microbes induce an innate response complement activation that trigger the adaptive response that is designed to eliminate these extracellular pathogens. Cytokine produced by cell during innate immune response to microbes or cell injury stimulate the proliferation of differentiation of lymphocyte in adaptive immune response. For example, cytokine secreted during innate immune reaction acting on B cell or CD4 T cell and CD8 T cell are given here. We have mentioned these cytokines previously, and we will discuss the detail of their roles in lymphatic response in later chapters. Interleukin-12 stimulates differentiation of naive CD4 T cell to a T helper 1 subset of effector cells and naive T cell to cytotoxic T cell. Interleukin-1, interleukin-6, and interleukin-23 stimulate differentiation of naive CD4 T cell to helper 17 subset of effector cells. In interleukin-25, interleukin-33, and TSLP stimulate the differentiation of naive CD4 T cell to T helper 2 subset of effector cell. Interleukin-15 promotes survival of memory CD8 T cell. Interleukin-6 promote the survival of antibody-producing plasma cell. Hope I said interleukin-15 promote the survival of memory CD8 T cell. Adjuvant are substances that need to be administered together with purified protein antigen to elicit maximum T cell dependent immune response. See chapter 6. They work by stimulating innate immune response at site of antigen exposure that promote subsequent adaptive immunity. Adjuvants are useful in experimental immunology and in clinical vaccine. Many adjuvants are experimental use and microbial product that engage toll-like receptor, such as killing mycobacteria and LPS. The most commonly used adjuvant in human vaccine is alum, which is composed of either aluminium hydroxide or aluminium phosphate, and may work by causing inflammasome activation. The CpG, the ligand of toll-like receptor 9, is used in some hepatitis B virus vaccine. Among the important effects, adjuvant will activate the dendritic cell to express more major histocompatibility molecules that are part of antigen signal 1, and T cell recognize increase the expression of costimulators signal 2 and cytokine needed by T cells activation and stimulate migration of dendritic cell to lymph node where T cells are located. Mechanisms that limit innate immune response. The magnitude and duration of innate immune response are regulated by a variety of inhibitory mechanisms that limit potential damage to tissue. Whereas the inflammatory response is critical important for protection against microbes. It has the potential to cause tissue injury and disease. Several mechanisms have been evolved to provide brake on inflammation, and these mechanisms come into play at the same time as shortly after initiation of inflammation. Furthermore, stimuli for initiation of many of these control mechanisms include some bump and dump that induce inflammation. We'll describe selected group of these regulatory mechanisms. Interleukin-10 is a cytokine that is produced by and inhibit the activation of macrophage and dendritic cell. Interleukin-10 inhibit the production of various inflammatory cytokine by activating macrophage and dendritic cell, including interleukin. Interleukin-10 inhibit the production of various inflammatory cytokine by activated macrophage and dendritic cell, including interleukin-1, tumor necrosis factor, interleukin-12. Because it is both produced by macrophage and dendritic cell, an inhibition of function of this cell, interleukin-10 is an example of negative feedback regulator. Alternatively, activated macrophage make more interleukin-10 than classical activated macrophage. Interleukin-10 is produced by some of non-hematopoietic cell types, e.g. keratinocyte. Interleukin-10 is also produced by regulatory T cell, and we will discuss detail of interleukin-10 in context of chapter 15. Loss of function of mutation in interleukin-10 receptor results in severe colitis developing in infants. Macrophage produce natural antagonists of interleukin-1 that is structurally analog to cytokine that bind to the receptor but is biologically inactive, so that it functions as competitive inhibitor of interleukin-1. It is therefore called interleukin-1 receptor antagonist. Synthesis of interleukin-1 receptor antagonists is induced by many of the same stimuli that induce interleukin-1 production. Some studies in interleukin-1 receptor antagonist deficiency might suggest that this inhibitory cytokine is required to prevent inflammatory disease of joint and other tissue, and rare disease caused by genetically deficiency of interleukin-1 receptor antagonists in humans is characterized by severe bone and skin inflammation. The recombinant interleukin-1 receptor has been developed as a drug for the treatment of rheumatic arthritis and familial fever syndrome, in which interleukin-1 production is dysregulated. Regulation of interleukin-1 mediated inflammation may also occur by expression of the type 2 receptor, which binds to interleukin-1 but does not transduce to activating signal. The major function of this receptor may be to act as decoy that competitively inhibit interleukin-1 binding to the type 1 signaling receptor. There are numerous negative regulatory signaling pathways that block the activating signal generated by pattern recognition receptor and inflammatory cytokines. Suppressors of cytokine signaling SOX protein are inhibitor of JAK-STAT signaling pathway linked to cytokine receptor. Toll-like receptor signaling in macrophage and dendritic cell induces expression of SOX protein, which limits the response of this cell to exogenous cytokines such as type 1 interferons. Pro-inflammatory response of the cell to toll-like receptor signal are negatively regulated by SHIP, an intracellular protein phosphatase that negatively regulates numerous thyroxine kinase dependent signaling pathway in lymphocytes. There are many other examples of kinase and phosphatase that inhibit toll-like receptor, node-like receptor, RIL receptor signaling, and small inhibitory RNAs that inhibit production of many of the mediators of innate immunity. In summary, the innate immune system provides the first line of host defense against microbes before adaptive immune response have had sufficient time to develop. The mechanism of innate immunity exists before exposure to microbes. The cellular component of innate immune system include epithelial barrier and leukocytes, the neutrophil, macrophage, natural killer cell, lymphocytes with invariant antigen receptor, and mast cell. The innate immune system uses cell-associated pattern recognition receptor present on plasma, endosomal membrane, and in cytosol to recognize structural called the pathogen-associated molecular pattern (PAMs), which are shared by microbes and are not present in mammalian cell, and are often essential for survival of microbes, thus limiting the capacity of microbes to evade the detection by mutating or losing expression of these molecules. In addition, these receptors recognize molecule made by host, but whose expression or location indicates cellular damage. These are called the damage-associated molecular pattern (DAMPs). Toll-like receptor present on cell surface and in the endosomes are important family pattern recognition receptor, recognizing a wide variety of ligands including bacteria cell wall component and microbial nucleic acid. The cytosolic pattern recognition receptor exists that recognize microbial molecule. These receptors include retinoic inducible gene (RIG), retinoic acid inducible gene (RIG) like receptor, which can recognize viral RNA, cytosolic DNA sensor, cytosolic DNA sensor, the CDS, which recognize the microbial DNA, and node-like receptor, which recognize bacterial cell wall constituent and also serve as a recognition component of many inflammasomes. Pattern recognition receptor, including toll-like receptor, node-like receptor, and the RIG, retinoic acid inducible gene like receptor, ARLs, signal to activate transcriptional factor, the NF kappa beta, which stimulate the expression of cytokines, stimulator and other molecules involved in inflammation, and the interferon response by factor, transcription factor, which stimulates the expression of antiviral type 1 interferons genes. The inflammasome, a specialized caspase-1 containing enzymes complex that form in response to a wide variety of pump and dumps, including recognition structure, which are often node-like receptor family protein, and adapter and enzyme caspase-1, the main function of which is to produce active form of inflammatory cytokine interleukin-1 and interleukin-18. Inflammasome-mediated proteolytic process of the cytosolic protein Gas-Demine generate membrane pores, which are conduit of release of interleukin from the cell and also cause osmotic cell death called pyrotosis. Soluble effect molecule of innate immune system are found in plasma and include the C-reactive protein, colectin, e.g. mannose binding colectin, and ficolin. These molecules bind to the microbial ligands, enhance clearance of complement by complement-independent, complement-independent mechanism. Innate lymphoid cells are cells with a lymphocyte morphology and function similar to those of T lymphocyte, but they do not express clonally distributed T cell antigen receptor. 3 helper subset of innate lymphocyte cells secrete the same cytokine as T helper 1, 2, and 17 helper T cell. Natural killer cells have cytotoxic function and secrete interferon gamma, similar to cytotoxic T lymphocyte. The natural killer cell defend against intracellular microbes by killing infected cell and providing a source of the macrophage cytokine. Interferon natural killer cell recognition of infected cell is regulated by a combination of activating and inhibitory receptor. Inhibitory receptors recognize class 1 molecule cytopropandability complex molecule, because of which natural killer cell do not kill normal host cell, but do kill cell in which class 1 molecular expression is reduced, such as viral infected cells. The complement system includes several plasma proteins that become activated in sequence by proteolytic cleavage generate the fragment of C3 and C5 proteins, which promote the inflammations, opsonize, and promote phagocytosis of microbes. Complement activation also generates membrane pole that kill some type of bacteria. The complement system is activated on microbial surface and not on normal host cell because microbes lack regulatory protein that inhibit complement. In innate immune response, complement is activated by mainly spontaneous on microbial cell surface or by mannose binding lectin to initiate the alternative and lectin pathway respectively. The two major effector function of innate immunity are to induce inflammation, which involve the delivery of microbes killing leukocyte and soluble effector molecule from blood into tissue, and to block viral infection of the cell mainly by antiviral action of type 1 interferons. Both types of mechanism are induced by pumps and dumps. Several cytokines produced mainly by macrophage, dendritic cell, and other innate immune cells mediate inflammation. Tumor necrosis factor, interleukin-1 and interleukin-1 activate endothelial cells, stimulate chemokines production, and increase neutrophil production in the bone marrow. Interleukin-1 and tumor necrosis factor both induce interleukin-6 production, and all 3 cytokines mediate systemic effect, including fever and acute phase protein synthesis by the liver. Interleukin-12 and interleukin-18 stimulate production of macrophage, activating cytokine interferons by natural killer cell and T cell. These cytokines function in innate immune response to different classes of microbes, and some, interleukin-1, interleukin-6, interleukin-12, and interleukin modify the adaptive immune response that follow the innate immune response. Neutrophil and monocyte, the precursor of tissue macrophage, migrate from the blood into inflammatory site during innate immune response because of the effect of cytokines and chemokines produced by bump and dumps stimulating tissue cells. Neutrophil and macrophage phagocytose microbes and kill them by producing reactive oxygen species, nitric oxide, and enzyme in phagosomes. Macrophage also produce cytokines that stimulate inflammation and promote tissue repair at site of infection. Phagocytes and recognize the response to the microbial product by several different types of receptor, including toll-like receptor, C-types, lectins, scavenger receptor, and N-formally made Leu-fen receptor. Molecule produced during innate immune response stimulate adaptive immunity and enhance the natural adaptive immunity response. The dendritic cell activates the microbes produce cytokine and costimulatory that enhance T cell activation and differentiate into effector T cells. Complementary fragment generated by alternative pathway provides second signal for B cell activation and antibody production. Innate immune response are regulated by negative feedback mechanism that limit the potential damage to tissue. Interleukin 10 is the cytokine that is produced by and inhibiting the macrophage and dendritic cell. Inflammatory cytokine secreted is regulated by autophagy gene product. Negative signaling pathway block the activating signal generated by pattern recognition receptor and inflammasome cytokines. Finally, this is the end of this chapter of innate immunity. It has been a very big chapter. Hope if you made follow-up, you learned something. And it's not necessary that you have all in your head. Rather, it is important that at least you have heard, you know where to find something that you need. Wish you all the best and best of luck as you're preparing for whatever you want in immunology. Thank you very much.